First of all, I would like to welcome everyone to the Soft and Biological Matter Session 1 in the oral presentation category. And I'm Kusha, and I'm from Barilan University, and I am the chair of this session. So first, we have two speakers here, Shiran. First one is you, and the other one is Sara from Italy. So before you guys begin your presentations, I would just like to mention a few points and a few rules that must be followed strictly. So for each presentation, you will be given exactly 15 minutes to present your work. And I will give you a heads up 3 minutes before the end of those 15 minutes. So please strictly follow this time limit. And because not respecting this time limit will deduct your points. Also, I'm really sorry, but I will be compelled to cut your presentation off if you exceed the 15 minutes time limit. After these 15 minutes, it will be followed by a 5-minute discussion or a question and answer part. And the attendees should reserve all their questions for this part. And you can put your questions either in the chat or just raise your hand and ask the question by yourself. Also, I would like to ask all the attendees to keep their cameras on because it will make the session a little livelier. But please make sure to keep yourselves muted, and unless you're asking the questions. So okay, so you can share your screen, Shiran, and amaze us. All the best.
Do you see the presentation?
Yeah, yes. Everything is okay. Yeah.
Okay, so I will start. So today I would like to present you my work on messy to order, the role of fiber diameter in determining the shape morphing behavior of 2D networks constructed of mesoscale responsive fibers. I'm from Tel Aviv University from Amit lab. So shape morphing materials and shape morphing systems have been studied a lot for different applications like soft robotics, artificial muscles, and also to learn and understand better the nature. And it has been done in many approaches. For example, Klein et al. Showed the fabrication of inhomogeneous sheets and its morphing into 3D structures. And Stoichev et al. Show the fabrication of bilayer sheets and its folding for 3D structures, and it has been done in many different ways, like 3D printing and photolithography. However, most of the systems that have been done so far are mainly is the bulk system. And when you want to make a morphing systems with the resolutions of only a few microns, it's much harder to achieve that. And for that, we use responsive polymeric microfibers, and we know that it works pretty well for biology systems, for example, to use these polymeric filaments to make hierarchical structures, and we know it works well for microtubules and actin networks, for example. So we actually want to know, can we fabricate this hierarchical shape morphing system using fibers? Will this structure affect the behavior of the system, and would it behave differently than a bulk? And for making this shape morphing system, shape morphing networks, we want to use the shape morphing material, and for that we're using the well-known thermoresponsive polymer. It calls polynipam, short of polyN-isopropyl-acrylamide. And when we take this polymer and its cross-links and we immerse it in water, the polymer becomes a hydrogel. It swells and absorbs all the water around it, and it expands. However, above a critical temperature of 32 degrees Celsius, the polymer collapses, it becomes hydrophobic, and the gel, as you can see in here, shrinks. And we take this polymer, we use this polymer, and we use the spinning method in order to make this really nice network structure. So in this method, we take the polyNIPAM solution with a cross-linker, and the solution is dispensed from a syringe and forms the fiber, and this makes the nice structure of a network. And with this, we can control two main parameters. The first one is the fiber diameter, and the second is the network density. So first, I would like to show you one of our results. This is a network with pretty thick fibers. Hopefully you can see it. Pretty thick fibers of over than 20. So the network was first at hot water when the fibers collapse, and then it swells and expands. And upon heating, now the polymer collapses into itself. And as a result of this, all of this network shrinks. And we call it the shape-preserving network. And this is really nice, but it's not such a big surprise for us, because we know that if we have inhomogeneous structure, we will get an homogeneous swelling, like we saw here and like we know from a bulk system. However, when we go into much thinner fibers, let's say this is another system of ours with 4 microns, and again, the network in hot water, and we cool down the water. So the fiber swells, and thanks to the voids of the system of the network, each fiber here can exhibit bending and buckling, and we call it the messy network, or the buckling governed network. And we see this is completely different than a bulk system, than a bulk morphing. And also upon heating, the network has this memory, and it goes back to this 2D Cartesian network, which is also pretty cool. So we wanted to understand what exactly is a messy network, when we get a messy network, when we get this preserving network, and for that we're using an order parameter. So when I want to check what is the fiber order parameter, I can take the fiber, divided it into vectors, and the sum of the vectors is actually the direction of the order of the fiber, and the normalized size of this sum is the size of the order parameter for one fiber. So for a network with lots of fibers, we do an average all over the order parameter fibers, and we did it for about 20 network, and in here, each black dot is one network, and this error bar is actually a standard deviation of the order parameters all over the fibers of the networks. So we can see here that for the thin fibers, we get this pretty lower order parameter, and this gets the buckling governed regime. However, above 7 microns, even a little bit more, we get this shape preserving regime when the network preserve its shape. So we're wanting to understand why this has happened, and we can look in here on the elastic energy of a filament. And we can divide this elastic energy to stretching energy and to bending energy. And we can see that for bending energy, the radius of the fiber goes like, the bending energy goes like the radius in the power of 4. So as the fiber radius increases, the bending energy increases more rapidly than the stretching energy, for example. And also the buckling critical force goes like the radius in the power of 4, and it also increases very fast. So we can understand that for thin fibers, the buckling critical force is pretty low, and the fibers exhibit mainly buckling. However, for thick fibers, we can see that we get this high buckling critical force, and the fibers exhibit mainly stretching. And we also wanted to make a simulation to understand even better our system, to know if this energy calculation and these mechanical calculations are true. So we use the spring-based numerical model in which we take our network and each fiber and divided it into a linear spring, as you can see in here, and the springs are connected by nodes or by angular springs, if you like. And throughout the swelling process, this numerical swelling process, we change three parameters. The first is the rest length increases, the radius increases, and the Young modulus decreases, and this is really happens also in our real systems. And we take all of this parameter and we plug it in into the bending and stretching moduli, and we calculate the force on each node, each angular spring, and we let the nodes move using their applied forces until a minimization of the force. So we did it for a range of diameters. And here are two examples. This is before the numerical swelling process. This is for 3 microns and for 10 microns. And after the swelling process, you can see that for 3 microns, we get this bending and buckling geometry. And for 10 microns, we get this shape-preserving network. So here we also calculated the order parameter for all the networks. So here are the results you saw before, the experimental results, and here is the simulation results, and you can see it fits amazingly well to our experimental results, which is also really nice. And we really think we understand the mechanical forces and energy of our network. So far, I talked mainly about the role of the fiber diameter, but what about other parameters? So we also wanted to check the density effect, the density of the fibers themselves inside the network. And we take one of the samples we check with the 5 microns. We did it like all the others, pretty sparse network. And this is upon heating, what it gets this Cartesian, 2D Cartesian network. And this is upon cooling when we get this buckling. But what happens if we won't allow the fibers to exhibit that buckling because they won't have the space to do this, because we restrict the fibers so much, because we make much denser fibers. So we wanted to check that, and we can see that for another network that was made with exactly the same solution, in the same spinning, with the same spinning process and the same spinning parameter, when we do a much denser network, and we again we pull it down, we see that this time we get this shape-preserving network, even though it's a pretty thin fiber. So it's not only the role of the fiber diameter, also the density help us to control the morphing of our system, which is also pretty cool for us. Okay, so I have the time, so I would like to conclude. So first I showed you how we fabricate these hierarchical shape morphing networks constructed out of these thermoresponsive polymers, thermoresponsive fibers, and show you how we control the parameters that actually changes our morphing behavior, which is both the fiber diameter and the network density. I showed you that for thick fibers and dense network, we get the shape-preserving networks, which is also a bulk-like behavior, which is what was expected from this network in the beginning with. However, that for a thin network with thin fibers and the sparse network, we get this messy shape upon swelling. However, it can still goes back to its 2D Cartesian geometry. And I also show you a numerical model that confirms the morphing behavior and the fiber diameter effect so far. And sometimes I've been asked, okay, this is really nice, but what are you going to do with all this? And we really want to learn how to control this morphing behavior and to make, let's say, like artificial micromuscle. And this actually happened by mistake, but you can see here that the network of the fibers acts as a muscle that moves this kind of finger. And this finger is about half micron thick and half millimeter thick and about 1 millimeter long. So it's really, even though the networks, the fibers themselves much thinner, they can still move very well this finger. And it's really nice for applications of artificial micromuscles, for example. So I would like to thank the Shulamit Aloni Scholarship and to thank my group and to thank all of you for listening.
Thank you, Shiran. It was a great talk and you finished right on time, which is awesome. Okay, so if anyone has questions, please ask. Yeah, Dana, you want to ask a question?
Yes, very nice work, Shiran.
Thank you.
I wanted to ask, how did you image all the video that you showed? It's in optic microscopy or in SEM?
Okay, so we're using the first samples, not the videos themselves. It was a SEM scanning microscope, electronic microscopy, but the videos and this, for example, is optic microscopy, and the one with black and white, let's say it's a fluorescent microscope, which is the same one, but there is another mode. But yes, it's optical and fluorescence microscopy in wide field and fluorescence.
Thank you.
Thank you.
Great. Any more questions? Okay, so I got a question. Okay, Iris, yeah.
Yeah, so I have two questions. The first one is, did you test it for several cycles for how reversible it is? How many cycles can it?
We checked it for a lot of cycles actually each time, and it's reversible. Actually, it's sometimes because of the water flows, because it's all in water. Sometimes if there is like flow of the water, then it becomes maybe a little bit messier with the messy fibers. However, it, let's say, dies when the water dries up. I mean, in the minute the water dries and it's on a slide, it's on a glass slide, and the fibers stick into the slide, can't move anymore. They still swell a little bit, but they can't move anymore, and then it doesn't work. But as long as the sample is wet and in water, it works pretty well for a long time.
I understand. And I have another question. You mentioned two kinds of for the some response for the thicker fibers, and some response for the more thin fibers. So are there any differences in the possible applications of the two types?
So we actually thought it would be, now we mostly characterize it and we want to understand it better, but we actually want to maybe combine these two. If we make, let's say, a network which half of it is thin and half of it is thick, and we get this messy part in one side and this shape-preserving network in the other side, maybe now we can somehow control something and maybe make this, let's say, kind of micro tweezers for that. We can do it, of course, also if we can control the density as well, and not only the thickness of the fibers. And yes, this is now, let's say, until now we wanted to check to characterize our system, and now we're starting the cool part where we can start to play with this and to learn how to control this and make maybe movements and stuff like that. But we've got a lot of idea for that. And we want to make, let's say, inhomogeneous networks and see what happens.
Very nice, thank you.
Thank you.
Okay, any more questions? Okay, I got one question. Can you move on to the experimental part of your, like the synthesis part?
Like which one?
Before, go a little more. Yeah, before that.
This.
Yeah, here. Yeah.
Okay,
so
yes.
when you show this syringe, it's like you're dropping it So it's not drop-wise, it's, I mean, what's the speed of dropping this on this? And there is there a spinning for it, or, yeah?
Okay, so the flow rate of the, let's say the drop rate, the velocity, it's pretty slow. It's about, and I'm not sure if it's called pretty slow, but it's 0.02 milliliter per hour. And so I get this drop actually, and I pull it down with a wooden stick. And because of this polymer is pretty big and it has this entanglement between the polymer chains, it forms this fiber, and I put it on this collector, this purple round thing. This is the collector, and this is continue to rotate. And the rotation makes sure that the fiber continues to form each time.
Okay, great. I got just one more question. Okay, you chose this responsive polymer, right? And but I just wanted to ask what inspired this work? Like, what exactly you wanted to do with it? Like you said, okay, you can go for a lot of applications, but do you exactly have something in mind, or if you want to share?
So we started with polynepam. You asked about this polymer exactly because it's a polymer that's also pretty easy to work with. All the systems I talked before, most of them are really using this polymer because it is a convenient temperature, critical temperature. So it's really nice. And we did want to, at the beginning, wanted to check what's going on when you make these hierarchical structures, but in the micro scale. Most of the systems that have been done before are in the micro scale, and we want to understand better what's going on when we get to this resolution. And we did inspired from the cytoskeleton, for example, because we know it moves pretty well. So of course we're not getting into nanometers of diameters like actin and intermediate filaments, but we thought that this haven't been investigated enough. This range of only few microns. We know that we have this really thin fibers of the biology. And we know that we have these really big networks with filaments of millimeters, but there wasn't anything in the between, and we really wanted to check what's going on in this range. And we get these nice results.
Okay, great. Thank you so much. It was a really nice presentation.
Thank you very much.
No problem. Okay, so let's move on to our next presenter, Sara Batista from Italy. I'm sorry, Sara, I cannot pronounce the name of your university. So
don't worry.
Go ahead.
Okay, so can I share my screen?
Yeah, sure.
Okay. Okay. Can you see my presentation?
Yeah.
Okay, perfect. So, good morning to everybody. First of all, I would like to thank the scientific committee for giving me the opportunity to show you my results today. This presentation will focus on the investigation about the physicochemical properties, antibacterial activity, and synergistic combination with antibiotics of structurally related liposomes. In general, liposomes are spherical vesicles having at least one lipid bilayer that incorporate an aqueous core inside them. They are biocompatible and very versatile aggregates because it's possible to functionalize them or changing their composition in order to modulate their properties. Liposomes are largely investigated as drug delivery systems because it's possible to encapsulate in them both hydrophobic and hydrophilic drugs. But they can also exert a pharmacological activity if devoid of an active principle. It's known that charged liposomes show a greater ability to interact with the cells, showing also pharmacological activity with respect to neutral ones. On the other hand, the presence of an oxide surfactant confers to these aggregates a low toxicity and a green character, besides an antibacterial and antioxidant activity as a function of their scaffold. In particular, the presence of a pyrrolidinium ring is peculiar because it can influence the hydration, the volume, and the topology of the polar head group, affecting the final properties of the vesicles. Liposomes under examinations were prepared by thin film hydration methodology and contain a saturated natural phospholipid, the DMPC, with one of the 4 structurally related synthetic alproinol derivatives showed in this slide. And the formulation differ for the molar percentage, the carbon chain length, and or the charge of the head group that can be cationic or zwitterionic of this component. All the formulations were characterized, and as expected, they show similar dimensions, about 80 or 90 nanometers. Zeta potentials showed some interesting differences. As expected, the cationic liposomes featured positive values as a function of their molar percentage and independently from the chain length. On the other hand, in liposomes containing any oxide surfactants, chain length influences zeta potential values. The relatively high value observed with the MPC C14 oxide formulations, despite the zwitterionic nature of these synthetic components, can be explained considering that the pyrrolidinium ring could be folded in order to expose the polar moieties, so the N-oxide and the hydroxyl groups, to the bulk, as observed in other systems containing analog surfactants. The folding could be also stabilized by the strong hydrogen bond between these 2 polar groups. The decrease of zeta potential values adding 2-methylenase in the alkene chain could depend on the presence of a chain length mismatch with the majority component, so between the majority component and the N-oxide derivative. The majority component in this case is the DMPC that bears 2 C14 chains. This characteristic causes variations in the properties such as bilayer thickness, fluctuations, and surface area in liposomes containing this kind of surfactants. Calorimetric data were obtained through differential scanning calorimetry measurements and give information about lipid organization in the bilayer. Results show that the inclusion of a synthetic surfactant in the MPC bilayer causes an increase of the van der Waals interactions, as indicated by the increase of the temperature of the main transition, the TM, and the decrease of the enthalpy variation associated to this transition. In the presence of C16 and oxide surfactant, we observed a broader peak than in the other cases about the main transition, and the disappearance.
Sarah, just a minute. Can you use some pointer or something? You know, because it's getting a little all over. Can you use a pointer to point out where you are in the slide? Yeah, that's better. Yeah. Okay. Start
okay.
again.
So to point to the transition.
Mm-hmm.
Okay. So I was talking about this is the main transition. This is the other transition. So the pre-transition. So in the presence of this C16 and oxide surfactant, we observed a broader peak associated to the main transition, and the disappearance of the pre-transition were observed, suggesting respectively a low packing of the bilayer and that the presence of a chain mismatch disturbs the arrangement of the polar head groups. These data are also in agreement with the zeta potential values that were the lowest in the presence of this surfactant. So all the formulations were tested on a panel of different microorganisms, such as gram-positive, gram-negative, and fungal strains, in order to determine their respective minimum inhibitory concentration, MIC values. I don't know if you can see this part of the presentation, so maybe I can do this better now.
Yeah, that's fine. Yeah.
Okay, okay. Okay, so surprisingly, cationic formulations did not exert any biological activity despite their relatively high zeta potential, often considered to the only relevant parameter in the interaction with cells. As expected, the best results were obtained with the gram-positive microorganisms due to the absence, in this case, of the external plasma membrane that didn't hamper the interactions between the liposomal formulations and bacterial cell wall. The best liposomal formulations were the ones containing the highest amount of the N-oxide surfactants, suggesting that the presence of this moiety plays a pivotal role in the interaction with the biological environment, independently from the zeta potential. It's reported in literature that also the lipophilicity of the chain can influence the antibacterial activity, but this phenomenon is not so simplistic, and it's strictly related to the nature of the microorganism. On the other hand, the inhibition was not observed at the investigated concentrations, neither for gram-negative bacteria nor for eukaryote Candida albicans, certainly depending in the first case on the presence of external membrane, and in the second case on differences in the lipid composition of the membrane. The antimicrobial activity shown towards methicillin-resistant Staphylococcus aureus is particularly interesting because of the multi-drug resistance of this phenotype. The strain is resistant to various categories of drugs, such as erythromycin, clindamycin, gentamicin, and oxacillin. So it was decided to test on this strain the best liposomal formulations, so the MPC C14 N-oxide and the MPC C16 N-oxide with the 20% of the N-oxide derivative in combination with these 4 antibiotics to assess the presence of a synergistic effect. It's important to highlight that the interpretation of this drug interaction with the tested liposome is influenced by the choice of the model of analysis. Among the several methods reported in literature, we used the FICI model based on the low additivity theory and the delta E model based on the Bliss independence theory. The first is more related to the MIC concept, whereas the second needs only the presence of a grow reduction to be applied. We observed a reduction of the effective antibiotic and liposomes concentration mainly in all the cases, but there were a synergy according to both methods only with oxacillin. In this case, liposomal formulations induce a drastic reduction of the antibiotic effective dose, about 16 and 32 times. Staphylococcus aureus becomes susceptible to a drug to which it showed a resistance. So this is a huge result. Oxacillin is a beta-lactam and inhibits the synthesis of the peptidoglycan layer of bacterial cell wall. This evidence suggests that the investigated N-oxide formulations strongly interact with bacterial membrane, creating a favorable environment to oxacillin to exert its action. Probably in the case of the other antibiotics that must penetrate into cell to exert their activity, liposomes not penetrate, or after penetration were disrupted by the biological environment and cannot act in a synergy way with the drug. So, basing on this result, the antimicrobial activity of the best liposomal formulations in combination with oxacillin was also tested against other Staphylococca, all resistant to this antibiotic. Specifically, we used the 2 coagulase negative species, Staphylococcus hemolyticus and Staphylococcus epidermidis, and a clinical Staphylococcus aureus. According to both methods, we observed synergies in all cases. In particular, for Staphylococcus epidermidis, the effective antibiotic concentration was reduced 8 times, falling downwards the breakpoint value. Also in this case, the strain becomes sensitive to an antibiotic to which it was resistant. So we can conclude that the results are certainly promising. So we prepared and characterized formulations that showed the ability to interfere with the cell growth, even if liposomes are not properly defined antibiotics. It should be possible to use them in adjuvant therapy, for example, in topical treatment, to improve the activity of antimicrobials or antibiotics such as oxacillin. The most important advantage is that liposomes don't have a specific target for interacting with the cell structure. They are chiotropic agents capable to determine morphofunctional alteration of the membranes, causing interferences with the function of the cells. The absence of a specific target is extremely important to avoid the possibility to develop resistance mechanisms using these detergents. In addition, the formulation containing any oxide detergents are substances with little or no toxicity, so their use in adjuvant therapy will be safe. As a whole, exploiting liposomes' property, it would be possible to achieve a dual function as drug carriers and antimicrobial agents. Further investigation to maximize the effect of each combination will be performed. In particular, analog formulation containing a higher molar percentage of any oxide synthetic surfactant will be prepared to evaluate the effect on the synergism with antibiotics and their efficacy also on gram-negative bacteria. I would like to thank all people who helped me in this work. At first, the research group in which I'm working in L'Aquila, especially Professor Luisa Giansanti and PhD student Elena Allegritti. Then I would like to thank Professor Luciano Galantini from University of Rome La Sapienza for the collaboration about dynamic light scattering measurements, and the research group of Professor Giuseppe Celenza from University of L'Aquila for biological studies. At last, I would like to thank again the scientific committee for this opportunity and all of you for your kind attention.
Thank you, Sarah. Yeah, we can have questions. If we have questions from the audience, please go ahead and ask.
Okay. Sorry, I have a problem with okay. Perfect.
Yeah, any questions from the audience? Okay, so I would like to ask a few. Can you tell us how do you make, I mean, what's the synthesis technique, if there is, like you're making liposomes and
yeah,
and you're like how you call it, dope it or like conjugated with the different ratios of carbon, I mean, can you explain it again?
yes, I can use a slide to explain better.
Yeah, exactly. Yeah.
This. Okay. Okay, this is the methodology of preparation of liposomes. So an amount of lipids and hydrophobic drugs were put in an organic solvent. The solvent was evaporated under reduced pressure to give a dry lipid film. This film, to this film was added a hydrophilic solution, in this case a PBS, a phosphate buffer. And after dehydration, the solution is stirred and heated to give multilamellar vesicles. After this part, we used to sonicate the solution in order to obtain a reduction of the size and of the lamellarity, in order to achieve small unilamellar vesicles that are it's possible to reproduce them easier than the multilamellar ones. So we use these small unilamellar vesicles to treat bacteria strains and so on.
Okay, so the hydrophilic drug and hydrophobic drugs, like you use both of them, or simply one and two, like simply one or the other? or I mean, so hydrophobic will be in the lipid bilayer, and the hydrophilic will be in the inside the core of the liposome.
In general sense, we can entrap both hydrophobic and hydrophilic drugs. So
Yeah,
the hydrophobic
right.
drugs will be in the bilayer in different positions. It's possible to study the positions through measurements. I don't know, for example, fluorescence measurements or calorimetry measurements. But it's also possible to put a drug inside in the aqueous core of the vesicle. But in this case, I talked about liposomes devoid of an active principle. So liposomes used as a kind of antibiotics, but also only for the composition. So these anioxide surfactants and cationic surfactants were in the bilayer, but not as a drug, as a component.
Okay. So probably you, so you use both of them like in the first step, like you show it
exactly here in this part of the, yes.
Okay, I understand. And what's the size range do you get?
The size range around 80 and 90 nanometers, so enough small.
Nice. Okay. And like apart from the antibacterial activity, what else you can, did you try like using instead of some antibiotics or some other like drugs for some other drug delivery application? Or you
with
plan
these
to
formulations, we have already an activity on bacteria strains, but we can try to add maybe an hydrophobic drug in the bilayer to try to treat gram-negative bacteria, for example. We are doing other studies in order to obtain an interaction also with strains that are difficult to treat. So this is possible.
Okay, fine. And
put together these two aspects.
Okay. And this is the only like method you use, and not any other method? Like, did you try other methods to do this, to make such a combination?
To prepare liposomes? No, this is not the only methods. It's possible to prepare liposomes, for example, by ethanolic injection. If, for example, you have some lipids that are not soluble in chloroform or solvent with the boiling, with a low boiling point, you can solve these lipids in solvents such as DMSO or ethanol, and in this case you have to use this ethanol injection. So you prepare a beaker with inside a PBS buffer. You have to heat this buffer, and after with a small syringe you have to have this injection of the lipid components, and you prepare liposomes in this way. But after, you have always multilamellar vesicles, so you have to treat by sonication or by extrusion or by other techniques to reduce the lamellarity.
Another technique, bat sonication or the probe one?
Probe one.
Okay.
Yes, you need a lot of energy to reduce the lamellar. So there are a lot of techniques to prepare liposomes, and each technique is more useful for a kind of aggregate that you want to prepare, or for the kind of components or drugs that you have to put inside. So it's possible to modulate the properties for achieve the target that you want.
Okay, thank you. So time is over. Thank you so much, both of you, Shiran and Sarah, for a nice presentation. And we have like now 10 minutes of break, and after that we will move on to plenary session 1. So see you guys there, and then we'll meet again in the next Soft and Biological Matter session 2. Okay, thank you so much. Bye-bye. See you soon.
Thank you.
Okay, yeah, fine. Okay, bye-bye.